A warm welcome for joining us at today's webinar. It's entitled Predictive Drug Discovery, Empowered with Cell Target Engagement. We're looking forward to a very exciting presentation today. We're going to bring together the two major elements in early drug discovery, predictive drug discovery and target engagement in cell. We have two excellent speakers today. You're not going to want to miss a moment of it. First of all, I would like to introduce Eric Sigel. He is a leader in the field of scientific computing, DNA-encoded library informatics, and data science. Throughout his tenure of 15 plus years, Eric has been involved with a number of drug discovery companies such as Zevi AI Therapeutics, XChem, and NVivo Pharmaceuticals. He is currently CEO and founder of Citadel Discovery, a company that offers solutions in the space of drug discovery AI. So welcome, Eric. We're very happy to have you.
Thank you.
We're also going to hear from Dr. Ivan Babic, co-founder at CellarisBio. He is an accomplished cancer researcher with a PhD in biochemistry and molecular biology from the University of Calgary. He has over 15 years of experience in drug discovery and has been actively involved in preclinical and clinical drug development. Dr. Babic holds patents for small molecule therapeutics and is the inventor of several novel drug screening technologies. So as you can see, we have a lot to look forward to today. We're just going to kick things off with some housekeeping. Today's presentation will take us approximately 30 minutes, after which we really want to hear from you all. So as we're listening to our speakers, when questions come up or comments, please pop them in the section in Zoom that's Q&A, the Q&A function. Submit your questions throughout the webinar, and at the end of the presentation, we will answer them. All right, now I am excited to turn things over to Eric. Eric, we're looking forward to hearing your presentation.
Thank you, Emily, for the nice introduction. Thank you, Ivan and Elmar from CellarisBio for inviting me to give this presentation. As Emily mentioned, I'm CEO of Citadel Discovery, also I would call myself an evangelist for DNA-encoded library machine learning. Citadel Discovery is focused on predictive drug discovery with DEL machine learning. So I'm going to go through a little overview of this field and how it applies to drug discovery. First off, AI has obviously gone through many hype cycles and AI winter cycles with changing from some periods where AI is going to save the world to it's going to destroy the world, etc. But I think we have finally reached a plateau of productivity where it's starting to actually produce some really interesting results. And that is a result of a number of things. First of all, the exponential growth in data. We're seeing an explosion of data in the social media space, Internet of Things, and of course, in our biotech fields, largely due to DNA sequencing capabilities. Of course, the compute power has massively increased as well, also on a steep upward curve, with AWS, for example, providing an opportunity for somebody at their home, in their home desk office, home office, to be able to access compute power that was unheard of in even large companies only a decade ago. And of course, there's the shift in algorithms. We've gone from simpler algorithms like random forests in past decades to deep neural networks and large language models that we hear about today. So how does this relate to drug discovery? Well, one of the biggest challenges we see for particularly small molecule therapeutics, where I'm focused, is an extremely high failure rate. We tend to be in this traditional approach of one molecule, or maybe better said, one series at a time. It's commonly stated that only 1 in 100 projects make it through the clinic and into to commercialization. But actually, if you go back a step and look at exploratory research efforts, we're looking at even worse odds, where it's 1 in 1,000 exploratory efforts leading to an approved molecule. And this is largely the cost and challenges here are largely baked in many cases, very early in the exploratory research and early discovery phases where we may not understand the biology well enough, may not have enough chemistry, and that leads us to compromises that may not be detected until well into the decade-long drug discovery cycle. And so the question is, how can we use machine learning to shift this? And I think we need to change our expectations a bit from how do we find the molecule? In other words, can we use AI ML to find a specific molecule that will make it through to all the way through to commercialization? I think that is, maybe decades from now we'll be able to do that, but actually what we really need to think about is how do we use machine learning to improve those odds of downstream success? And a couple of ways would be to create early on, this early stage, more starting points with better drug-like properties, so that they won't fail later in the cascade. So if we look at this in terms of this cartoon I showed before, what we're trying to do is shift from that 1 in 1,000 to many in 1,000 that ML could generate, starting with lists of hundreds of molecules with therapeutic potential that can be then filtered by the ADME tox and drug likeness properties and increase the likelihood of success downstream. So how does DNA-encoded libraries impact this new thinking and that is through the use of DNA-encoded library machine learning. So I led a project when I was at XChem along with some of my coworkers at Google Accelerated Sciences using DNA-encoded libraries, or DELs. For those of you who aren't as familiar with DELs, the idea is that you create a combinatorial library where you grow the molecule using building blocks, but simultaneously using an attachment or headpiece molecule, you add DNA barcodes that encode for those molecules. And by having these, by creating libraries in this way, you can create libraries that are anywhere from hundreds of millions or even billions of molecules. What we did was we took a stream of a number of targets of interest. We fed this DNA-encoded library data, which can be which can be in the hundreds of millions, tens of millions, or hundreds of millions of data points. And we use those in a random forest model training and GCNN, or graph convolutional neural network training, to create a predictive model that allowed us to then take large-scale collections of molecules and predict molecules that we then acquired and assayed. And the top line result of this work that we did was that all 3 of the targets we tried, and we only tried these 3 before we published, we had significant hit rates. And these were various target classes. Overall, we had 30% of the tested compounds with a 30 micromolar or better potency. We did see that the graph convolutional neural network did outperform the more simple random forest, but actually random forest did quite well and provided different insight into different chemistry. And the other thing that the GCNN was able to do was to demonstrate higher potency, or sub 10 nanomolar potency hits for all 3 of these targets. I have a couple of IC50 readouts here just to show that these were not artifactual results. A couple of characteristics of those results that I want to point out. One was we were able to take these models and apply them back to known chemistry for the targets that we were interested in. And we were able to show that the model was able to enrich in the top percentile a considerable amount of that known chemistry. So a really good sign that the model is working. But we were also able to show that we could find novel chemistry. And here we're looking at average Tanimoto score of each of the hits we found to each of those 1,857 chemical actives, and the maximum Tanimoto score. And you can see that there are a number of molecules that were quite dissimilar from the known molecules. Additionally, those models generalized well beyond the training data, and we were able to show for all three targets, in fact, some of the most potent molecules were quite dissimilar or significantly dissimilar from the input molecules, whether positive or negative training examples. And finally, we wanted to show that this model wasn't just simply a really expensive way to do a similarity search. And so we took the machine learning out, replaced it with a similarity mechanism, and we were able to show that that actually led to no hits, whereas the random forest and deep learning did quite well. So there's something more to it than just similarity. We also were able to show that you can use these models in optimization phase. We took 6 of the series, or 6 of the molecules, seed molecules in series that we discovered in that first round of machine learning, and we were able to use a relatively high similarity plus high prediction score to generate follow-up molecules, where we're able to increase the potency of all 6 of those series by anywhere from 8 to 40-fold with 100 molecules, 100 off the shelf, or in that case, I think enamine real molecules. We also showed that you could take some of these modestly potent molecules, 1 to 10 micromolar range, and follow those up with even just a small number, about 5 follow-ups each. We were able, for 20 of these 23 seed molecules, we applied again a relatively similar 0.45 Tanimoto score ECFE4 and a high prediction score, and we were able to jump potency for almost half of these 23 molecules, anywhere from 3 to 60-fold potency in just $500. They were $100 per enamine real molecule at the time, so for just $500. So we're able to get more starting points, and that gets back to the point I made before, where more starting points can lead to fewer compromises. We were also able to show that these models could actually apply, in this case retrospectively, to an optimization campaign where the predictions were stronger for molecules that were active and less strong for the less active molecules. So does this approach generalize beyond the initial proof of concept? In fact, many groups have publicly disclosed ML efforts. This slide is from a while ago, and there's more. There are more groups, but we've seen dozens of targets and actually at least more than 10 different separate DEL library sources that have led to successful projects. So from those two aspects, it definitely is working. And then there's been a number of different publications in the last couple of years on new targets, showing new target application to new targets, showing application to designing DEL libraries, different model types, to the combination of DEL machine learning and computational chemistry, drug property prediction, and even cell-based selection. One of the key problems, though, that I see right now in the industry is we do not have the infrastructure set up to drive this cycle of machine learning and drug discovery. And with timelines that take 2-3 months to, in the best of cases sometimes, to get molecules for testing, this is far too slow for machine learning to compete with the much faster cycling that medicinal chemists can do with one-off molecules. So we really need to adjust this, the industry capability to drive rapid chemistry generation and biological testing. So if all of this works, and we're obviously seeing signs that it's working, we could take this approach and, for example, apply DEL data generation across many different proteins in a particular disease pathway or disease space. And so here's an example using lung cancer proteins, where we could generate data for all of these and then potentially build a whole pipeline of candidates candidate starting point molecules, and then create opportunities to drive whole disease area focus using this kind of approach. So now what? And I was thinking about the dog catches car concept. What happens when this all works? What happens when you have increased the number of series for a single target, and you've generated a number of series maybe across multiple targets? This is a challenging problem, but a good problem to have. And this is where I see the CellarisBio and Citadel Discovery work could potentially join forces, which is now you can take those molecules and begin to triage them if you have a higher throughput approach to looking at cell target engagement to make sure that you're actually pursuing the molecules that you want to pursue based on their mechanism of action. So thinking big, what are some of the next steps? Well, first of all, I think DEL can be used to generate data at the proteome scale. In other words, thousands or even multiple thousands of proteins. In fact, some of the DEL providers have already pushed into that space of thousands of proteins, and that could create the kind of data volume that we see enabling the ChatGPT large language models, but in this case on protein and ligands. We also are starting to see capabilities and approaches where protein sequence is being used to predict what ligands to pursue without any wet lab work. I personally think there's probably not enough data out there to do it, but I think DEL as a source of that data actually could provide a really interesting approach, enabling a true understanding, broad understanding of ligand and protein interactions. The third box here is greater access to target to tool molecules, etc., will enable target validation on a much larger scale. So getting this data into the hands of academics would be a really huge benefit to the industry and to human health in general. And finally, as more data sets are generated across pathways, across target classes, can we then use that, and I believe we can, to create more complex chemical biology models for understanding disease spaces. So with that, I just want to thank the XChem team, Zebi AI team, the partners that I mentioned, and of course the Google Accelerated Science team in that original research, and my team at Citadel Discovery, and of course CellarisBio for having me. And with that, I can turn it over back to Emily.
Thank you, Eric. That was fascinating. We really appreciate you sharing your experience and knowledge with us. I loved the dog catches car question. What now? That was fantastic. All right, now we're going to turn it over to Dr. Ivan Babic. We're really interested to hear what CellarisBio can contribute to this conversation.
Thank you, Emily. Thank you, Eric. So what I'd like to talk about today is how to de-risk drug discovery and development programs, and specifically with a focus on incorporating cell target engagement at the forefront of drug discovery programs as a way to de-risk these programs. So when thinking about de-risking drug discovery, some of you may be familiar with these curves. On the left is a graph that shows increased R&D spending over the course of about 35 years from 1980. And on the right is a graph that shows the number of drugs that are actually produced per $1 billion spent in R&D. And what's really striking is the inverse correlation of this. And what we're looking at is a failure of the drug discovery pathway to develop cost-effective drugs. So a failure to develop, a single drug in a way that you decrease the amount of spending on R&D. And so the question is, how do you reverse this trend? And so, first of all, why is there this decline in productivity, and how can we improve upon this? So when thinking about targeting diseases, we start to realize that there's a biological complexity of a lot of the diseases that we're looking at or trying to study, and specifically looking at targeting proteins that are drivers of some of these diseases. And a lot of these proteins function in the biological complexities of the cell. And so when trying to develop assay systems to assess drugs that can modulate the activity of these proteins outside of the cell, you're dooming the program to either stall or fail. So in order to reverse this trend and make it much more cost-effective in terms of developing drugs and improving the productivity of drug discovery, I think AI ML, as Eric mentioned, will help to improve that productivity. But also at the same extent, we need to introduce tools that could help in terms of identifying potential therapeutics to important disease drivers, and these tools that maintain the biological complexity within which these targets function. At the heart of this, cell target engagement should be at the forefront of some of these drug discovery efforts, because it's cell target engagement which maintains biological complexities and which can help guide some of the important decisions early on in the drug discovery programs. So there are two ways to de-risk drug discovery and development. The first would be to address target validation. This is absolutely critical because the majority of drugs that fail in the clinic is due to the lack of efficacy. And the underlying cause of this lack of efficacy is a poorly understood and validated target. And this is at the early stage of drug discovery, and most of the research efforts around target validation are conducted in academic research labs. And as the focus moves towards the translational research, towards developing a therapeutic targeting a specific drug, it's important that we spend more resources and time and increase scientific rigor around target validation, because if early on you don't have much biological understanding of the importance or relevance of a target to the disease, and further on down the cycle, you'll start to get stalled in your discovery path for drugs that could potentially be therapeutics for the program and potentially fail in the clinic. So another way to address or de-risk drug discovery is through incorporating target engagement early on in the drug discovery path. So after a target is well validated, by incorporating cell target engagement strategies as a way to identify potential modulators of that particular target is a good way to de-risk drug discovery programs, because these types of assay systems or screening systems maintain the biological complexity within which some of these targets that we're trying to drug are actually functioning. And so this is especially important for these challenging targets that cannot be expressed outside of the cell. And so it's important to incorporate these kind of strategies early on that could help guide important decisions in the drug discovery campaigns. And so why is cell drug target important? Well, as mentioned, in order to identify molecules that could modulate the function of a well-validated target, it's important to maintain the biological complexity within which that particular target functions. And having cell target engagement early on in the process of drug discovery helps incorporate a translationally focused program that could guide medicinal chemistry efforts and provide physiologically relevant data for these decisions. As Eric mentioned, combining a way of quickly triaging potential hits that come from an AI ML screen is a good way to seamlessly speed the drug discovery efforts in a way they can also de-risk further on down the pipeline. So another reason to incorporate cell target engagement and as a means for de-risking programs is looking at challenging drug targets. So the human proteome has about 18,000 proteins, and about 4,000, just more than 4,000 of these proteins are actually disease-related proteins or have some underlying function within the course of a disease. And we've only drugged a small fraction of these proteins. Now, the remainder of the proteins remain difficult to drug, and they're considered challenging targets. And so what makes them challenging is some of these features on the right here. So for example, they may lack a hydrophobic pocket that's potentially druggable site. They could be functioning as multi-subunit complexes. And most importantly, many of these challenging targets, they lack a 3D structure when you express and purify these outside of the cell, which makes it impossible to synthesize these recombinantly and have purified protein to work with. And so it's for these challenging targets that cellular drug target engagement should be at the early stages of drug discovery. Now, there are two ways to address target engagement. The first is a biophysical way. And so for biophysical methods, you require a recombinant protein. And typically these are used in an artificial environment where the purified protein is combined with a potential ligand on a surface of a microchip. And so there are various ways to look at this interaction, for example, SPR, ITC, BLI, and some of these have been around for over 30 years, and they work great for these easy targets that can be expressed as recombinant proteins. But as we're learning about biological complexity of some of these diseases and the importance of maintaining a physiological environment when screening for modulators of a function of a disease driver, we need to make focus cell-based assays specifically for target engagement. As these maintain the physiologically relevant environment, they're appropriate and perfect for these challenging drug targets. And importantly, they're also amenable to multiplexing as well as multi-omic analysis, which can be useful. At CellarisBio, we've developed a microtag cell target engagement platform specifically designed for these challenging drug targets and specifically designed to help de-risk some of these drug discovery efforts because it's designed to maintain a physiologically relevant whole cell environment when screening for potential binding molecules to particular targets. The platform, we use real-time detection instruments such as qPCR machines and live cell imaging. So this offers a greater dynamic range, increased sensitivity by incorporating this platform into these instruments. So the platform itself is an end-to-end platform and can be used at various stages in the drug discovery process. So we can incorporate this platform early on in the discovery phase where we can use this platform to look at DNA-encoded libraries to identify potential chemical matter or new matter that can potentially bind to a particular target. The platform was ideally developed for ranking potency of binding of molecules within a challenging or biologically complex environment within a whole cell environment. And we can also use this particular platform to look at mechanism of action of some of these targets because it's monitoring the interaction of a drug with its target within real time, within biologically complex systems. So either live cells or lysates derived from cells. CellarisBio has collaborated with Thermo Fisher to develop a novel strategy for a cell target engagement that can look at direct binding of a drug to cells using a qPCR machine. And so this is a simplified model of what's required for this is just basically a microtag version of the target expressed in cells. The cells are then transferred to a PCR plate and combined with a potential inhibitor or ligand with detection reagents and immediately placed into a qPCR machine and monitored for fluorescent signal with increasing steps of heating. And the type of readout or output you get from the system is shown to the right here, where a DMSO vehicle control, you start to lose fluorescent signal because you're increasing heating, which causes denaturation of that target protein. In the presence of a potential ligand that will, or potential binding molecule to the target, you start to see an increase in fluorescent signal, which is from stabilization of that particular target within a heat denaturation. Here's a case study for a G protein coupled receptor. So G protein coupled receptors are challenging targets in that they cannot be expressed outside of the cell and they require the biological complexity. And in this case, taking cells expressing the G protein coupled receptor and combining it with a test compound and the detection reagents, putting it into the qPCR machine, programming these step gradients, what we start to see is this separation of the fluorescent signal in the presence of the test compound at about 45 degrees Celsius for this particular protein. And this separation, again, is because under the vehicle control, you're starting to get denaturation and loss of that target protein. So you're not going to get a fluorescent signal being generated. But in the presence of a potential binding molecule, that molecule will impart a thermal stability to the protein, which allows for the generation of a greater fluorescent signal. Another case study here with an enzyme called ubiquitin conjugating enzyme. So this particular protein is challenging in that it functions as a multi-subunit complex. It mediates transcriptional activation of target genes. And again, in the same fashion, although this particular case was using non-denaturing lysates that were prepared from cells expressing this particular protein. So taking these lysates, combining it with a ligand and combining it with detection reagents and then monitoring fluorescence signal in the presence of the step gradient, we see the separation of the test compound from the vehicle control at about 50 degrees in this particular case, suggesting that there's binding event between this test compound and the protein target of interest, which stabilizes the protein in this thermal step gradient. Now we can take these lysates and perform a dose response, and in this case, from 4 micromolar down to 40 nanomolar. So as you increase the amount of the ligand in this case, you start to get the separation of this test compound at about 640 nanomolar. Now, below that concentration, the curves overlap, which suggests that there's no binding taking place between that compound and the target protein. But above 640 nanomolar, you get significant separation of the two curves, suggesting significant binding has taken place. And we can quantify this in-cell target engagement in this manner by taking the area under the curve between the test compound and the vehicle control. And then we can derive an in-cell KD affinity measurement by plotting the inhibitor or dose concentration of the ligand versus the area under the curve. And in this case, we can derive an affinity constant of about 1.2 micromolar. So this is a great strategy that has been developed to identify an affinity measurement for a drug with its target in a complex environment. So just to conclude, some of the things that were touched upon today. So the predictive power of AI ML can help increase the success rates in therapeutic drug discovery. So at CellarisBio, we've developed a microtag cell target engagement system that's ideal for assessing the binding of drugs to challenging drug targets. This is amenable to high throughput systems. We employ real-time instruments, which provides a high level of sensitivity with a high dynamic range. And this platform for target engagement offers streamlined workflows and is much more, I guess, advantageous compared to other conventional means for target engagement. And I think that's it. Thank you very much. I'll pass it back to Emily.
Thank you so much, Dr. Babic. It was really interesting to learn some of those challenges and the very exciting solutions that we have available to us. All right, guys, so that includes the presentation, but we would love to hear any of your questions that might have come up during this that you'd like to address either to Dr. Babic or to Eric. We have a couple come in already, so we'll get started with those. But if while we're talking, more come through, please feel free to pop them either in the chat or the Q&A function. So if we could start with Eric, one question that came in quite early in the presentation was about traditional QSAR models. Do you think that those models built with random forest are still of any value, or have we now transitioned exclusively to the AI ML models built using deep neural nets?
Yeah, so It's interesting. I think there's a lot of hype around deep neural nets, and I think they work quite well in a lot of cases. But I have certainly seen random forests outperform on many of the Citadel projects I've done. So from that perspective of just the model random forest versus neural networks, absolutely, I believe random forest is still very interesting direction. The other thing, as I mentioned before, random forests and GCNNs, in the case of our work, produce different results. So we actually got different chemistry out of the two. QSAR models absolutely still think they're relevant. I think they will be even more relevant if we can increase the amount of data that we can generate by making higher throughput analyses. But I think the sort of fundamental of QSAR is that it's more often used in a series, and once you have enough data in that series to develop your molecules, whereas the DEL approach and the AI/ML approach that I was discussing allows you to operate without having all of that data up front.
Fascinating. Thank you, Eric.
Absolutely.
Wonderful. Perfect. Can we move on to Dr. Babic? We have a question come through for you. Ivan, what can you say about how you configure this method for deconvoluting targets?
Sure. So with this particular method, it's a great way to assess binding in a high throughput manner. So if for example, you have a number of potential targets to a compound, you can quickly test which one of those targets off of a list, let's say of a mass spec proteomic profiling that you've done. We can quickly identify which of those bind to this particular compound and rank potency of binding of those. We can also help you in terms of starting with deconvolution of your particular target. So many people have drugs that they identify from functional screens, and then one of the issues of that is you don't really know what targets that particular drug is binding to impact the function or the readout that you have. So what you can do is there's various strategies. There are total proteomic profiling that other companies can do. Or what you can do is using tags for the compound of interest. If you can tag that particular compound, do a pull down, send for mass spec, identify potential targets. The use of this microtag platform can quickly identify which of those targets it's binding to and can rank these in terms of potency.
Fantastic. Thank you, Ivan. Okay, one more for you. Ravi says there are many parameters that affect the ligand binding. How do you monitor those parameters other than the temperature?
Sure. So with respect to the assay itself, we're using temperature as a way to force the denaturation of a target protein. So as you increase temperature of any protein, they'll start to melt. Hydrophobic amino acids get exposed to the cell surface, and so you'll get this denaturation, aggregation. The protein becomes insoluble, and then it can no longer function with our detection reagents. So that's the basis. It's a protein thermal shift type of assay. There are certainly many parameters that can affect ligand binding. You're looking at buffer conditions, salt conditions, pH conditions. So this is a reason why we want to employ whole cells, right? It's the whole cells, the physiologically relevant environment that we're assessing that binding of the drug to its particular target in that physiologically relevant environment. And so we are not playing around with any of those conditions. It's the conditions within which that target actually functions that we want to maintain. The only parameters that we adjust is temperature to try to force that denaturation of that target, and that denaturation can be rescued by the binding of a ligand. That's how we can detect it.
Very interesting. Okay, I think we have a follow-up from Stephen here. Can you do shield plots in the cell?
That's something that we haven't done yet. that's something that we can certainly explore.
Okay, fantastic. All right, maybe we can pop it over to Eric if you could answer a couple of questions that came in. The first one is, what are the factors for choosing a Dell library for a successful drug discovery project?
Yeah, I mean, from my perspective, the most important factor for choosing a Dell library is to make sure that you can get all of the data. So a lot of the a lot of the providers are sort of shifting into this direction of providing more and more data, but without that data, you're kind of limited as to what you can do. I think personally, I think that there's benefit in large diversity libraries, but I don't think it needs to be the most massive Dell library you could possibly access. In fact, I would certainly 100% take data access over increased diversity any day. So I think a well-constructed Dell library, particularly solution phase Dell, XChem, HitGen, Wuxi type of libraries, those are providing strong diversity for machine learning purposes. Beyond that, I don't particularly have a sort of a proclivity towards specific libraries for specific targets. I do think there's kind of the next step library that you could consider. Once you've done that first exploration of your target, you might want to create a targeted Dell library, particularly if you can use the machine learning to derive that library.
Okay, great. Wonderful. One more for you, Eric. Can Dell discovery be conducted within the complex environment of live cells?
Yeah, actually, there have been some very interesting experiments. Vipergen in the last couple of years published an article on their use of performing DNA-coded library selection in xenopus eggs, or very large cells, and performed in there. I do think this is something that companies are really interested in doing. I think it is possible. I don't think we are necessarily approaching its sort of regular application yet in industry, but it is definitely something that can be done, but needs some more development to become more regularly used.
Exciting future. Fantastic. I'm not sure who would be best qualified to answer this question. Mina asks, how do you calculate the binding affinity?
Sure, that's something I can address. So with respect to the binding affinity, what we're doing in our assay system is we're combining the ligand with the target or the cells of interest that are expressing the target. And this is prior to any sort of equilibrium has been reached. So just combining these together and monitoring fluorescence development, which is derived from the amount of derived from binding of that particular ligand to its target. So it allows us then to plot dose of inhibitor versus fluorescent signal, either raw fluorescent signal or area under the curve. And from that plot, we can identify an in-cell KD or derive an in-cell KD. It's affinity measurement that's for this complex system.
Okay, fantastic. And from George, what possibility is there that we can improve on ML to actually determine ADMT in silico?
Yeah, so I can take that one on. I think that the machine learning is probably not the issue. I think our methods are probably outpacing our data collection. So I think one of the major things in terms of ADME-tox in silico predictions, I think what we need is access to data sets that are more broadly available. There's been a lot of approaches, and actually some of my contacts have started a company and have been very successful in bringing this data in a sort of consortium manner across multiple organizations to create the sort of state-of-the-art molecules in ADME-tox prediction. But of course, you have to be part of that consortium or potentially pay a lot to get there. the other piece is how well are those data sets playing together? So I think one of the key elements here for improving ML in ADME tox scenarios is how do we create more consistent data sets? And one of the things that's beneficial, for example, about DEL is it's the same experiment for all of these molecules. How do you create consistent data so that you don't have this sort of data aggregation, um, from different sources confounding the system? So I think there's logistic and scientific questions that on how to create data sets that are bigger and better quality is the main thing in those to answer that question.
Lots more to explore. Fantastic. All right, we had a couple of questions about the microtag cell target engagement platform. So Dr. Babic, do you need to label the compound for that platform?
So when using this platform, the only thing that would be labeled is a 15 amino acid tag would be clonally added to your target. The compound itself is not labeled, so the fluorescent signal that we're actually detecting is through our detection reagents. It works through enzyme complementation, but the compound itself is not labeled, so that's label free.
Okay, fantastic. And also regarding the microtag target engagement platform, can it be used with transcription factors?
So transcription factors are a very challenging drug target, right? Because these are difficult to express outside the cell. Many of these behave or function within multi-subunit complexes. So trying to explore or identify modulators of their function outside the cellular environment is not going to work. So this type of platform is perfectly designed to look at drug discovery programs that are specifically focused on transcription factors. We have a couple of case studies that we've worked on. So MYC is one transcription factor that we have a case study that for this particular type of target engagement strategy.
Okay, fantastic. That takes us to the end of all of our questions. Thank you, everyone in the audience, for participating. Thank you for your excellently well thought out questions. And thank you so much to our panelists. This was absolutely fascinating. So, all of you in the audience, if you have any other questions, please email us. We have our email address there on the screen. It's hi, at celarisbio.com. You can also follow us on LinkedIn and our website, solarisbio.com. Thank you to our panelists, to all who participated, and please stay tuned. We have future webinars coming up that are very exciting and informative. So we'd like to wish you all a wonderful day, and we'll see you next time here at Solaris Bio's webinars.